As we begin our discussion about animals in our tree of life, we're actually going to start with a few characteristics of animals today. Now, I know many of you are familiar with some of the developmental properties in animals, but I want to review them very briefly just to make sure that we're all on the same page. So we'll start eventually our discussion with invertebrates. This is one that always strikes folks' attention. This octopus on the slide is a blue-ringed octopus, and typically we look at the octopus as having defense mechanisms based in camouflage, but this one is an exception. It actually produces a toxin that's 10,000 times more lethal than cyanide. So it's a pretty cool organism, and we'll look at some really interesting features in animals when we go through. So in general, we look at animals as being multicellular. They're going to exhibit different types of embryonic development that we will look at after a bit. We also need to look at how they eat. So they are going to be heterotrophic, meaning they have to eat their food. They cannot photosynthesize. And when we look at them, we're not going to talk about cell walls anymore. Those are plant characteristics and fungal characteristics. Instead, we need to look at how they're going to develop so we can understand how they're related, so comparative embryology. And we look at most of them as having some sort of internal digestion, but we'll look at a few invertebrates that do weird things that are more similar to the fungus where they have external digestion. Most of them are going to move around. We have a few exceptions. Sponges don't move around much, so we'll look at those. We look at all of them as being organized into tissues, so we're going to have to take that multicellular feature, build tissues, and then organ systems. Their symmetry is variable. Their symmetry is how they can be divided out, so we'll look at different types of symmetry there, primarily radial and bilateral. Okay? When we look at the whole kingdom Animalia, that would include all of our invertebrates and vertebrates, we're actually talking about 36 to 41 different phyla, depending on how you categorize them and depending who you talk to. Now, if this is kingdom Animalia, remember that we had a domain above that. Do you remember what domain we're working in? These are all eukaryotes. So kingdom Animalia, and then after kingdom we divide things into phylum. So we will look at 9 different phyla as we go through our invertebrates and vertebrates. And finally we will end up with chordata, which is the chordates, and the ones you're most familiar with, fish and reptiles and so on. But we have 8 others to get you familiar with first. So when we look at a body plan in an animal, we have to think about how it's divided out, so its symmetry, that radial and bilateral that I mentioned to you. We'll also look at whether or not these organisms actually have what we call true tissues, meaning that they're setting up higher complexity development so that they can be higher complex organisms in the long run. Number of embryonic layers, we'll look at whether an organism has 2 or 3 embryonic layers. We've talked about these before as germ layers. Okay. This body cavity will become important. This helps us divide out the body internally and helps us protect critical organs. And then how the development in the embryo actually occurs. Some of you may remember these as deuterostomes and protostomes from when we actually talked about development. So there's huge diversity in the animal kingdom. It's probably not a surprise to you. We'll look at all of these different organisms as all being animals, even this one in the middle that looks more plant-like, still an animal. So we'll look at that.